Okay, the next topic on the chemistry of life are about enzymes. So enzymes, they of course they are very important because a lot of chemical reactions that are occurring in cells, in living cells, involve enzymes. So the reason why there is an effective chemical reaction within the cells because they use enzymes. So okay, under enzymes we have to include their importance, okay, importance and their uses. Okay, so in the case of importance and uses, just like what I said a while ago, all cells carry out chemical reactions by using minimal amount of enzymes, and by using enzymes, enzymes are able to change a specific substance into another substance and speeding up its rate of reaction. So right after that, of course, you have to write down the definition of enzymes. Definition being the enzymes are actually being used as biological catalysts. Catalysts because they speed up the rate of reaction. And when enzymes are being used along the process of the chemical reaction, they themselves as enzymes are not being changed along the process. Okay? And they're also made up of proteins as well. So right after the definition of enzymes, okay, we have to introduce what active sites are. Okay, active sites. So in the case of active sites in enzyme molecules are a specific crevice or a specific space in the enzyme molecule where the substrate can bind. Okay, so this is where the chemical reaction can occur within the enzymes. So right after that, define what a substrate is. Substrate, this is the starting material that is being used in the chemical reaction. That is going to be changed into a product later on, okay, by the enzyme. So right after that, we have to indicate this this area later on. So enzyme substrate specificity. Okay, so when we discuss the area under active site, we can also discuss that these active sites are only specific for their substrates. Okay, meaning each enzyme can only work with a specific substrate. So by this condition, then we are able to have this enzyme substrate specificity. It actually works according to lock and key hypothesis, wherein the enzymes, whenever the substrate attach rigidly onto the active site of the enzyme molecules, and as they attach on the active sites, they undergo an enzyme substrate complex. So under this enzyme substrate complex, there are certain chemical bonds that are being broken down on the structure of the substrate molecule. So when bonds are being broken down in the substrate molecule, it leads to the production of a product later on. Okay, but under lock and key hypothesis, we can also clear out that this lock and key hypothesis is not really, we cannot really compare the activity of enzymes under this, because enzymes, they remodel themselves later on as they undergo the chemical reaction. Okay, so under the induced fit model, the active site of the enzyme molecule can break down certain bonds, chemical bonds that are present in the active site. So it can fit properly, it can fit like a glove with the substrate, which increases the enzyme activity of the enzyme itself. Okay, so we can also include chemical equations. Okay. So under chemical equations, we can say that whenever substrate A whenever substrate A is going to be attached to enzyme B, then later on it's going to be produced as the product C, and of course the enzyme B in the end. Meaning the enzyme along the way, it doesn't get changed. Okay? So, and also the chemical reaction is also reversible, most of them. Okay? So we can also include diagrams under this area. Okay, so in diagrams, by using diagrams, we can further explain to the students the idea under substrate and enzyme specificity. Like when, for example, whenever we have a specific substrate, okay, so this structure, okay, so this structure of the enzyme, of the substrate, sorry. So for example, if this is the enzyme and then this is the substrate, as you can see here, that this preface is only specific for this specific substrate. Okay? Meaning if there are no chemical bonds or chemical properties present here in the substrate that doesn't fit the chemical properties here in the enzymes, then the substrate wouldn't fit. Okay? So right after that, okay, right after explaining this enzyme substrate specificity, we can include denaturation. Denaturation, this is actually an event or a specific chemical reaction that occurs in most proteins, wherein there is a chemical change that is happening in the protein, wherein there is an alteration in the structure of the protein. So whenever an enzyme is going to be denatured, it loses its biological property in changing the substrate into a product. Okay? So, chemical change. So last but not the least, right after explaining what denaturation is, we can now explain the factors affecting enzyme activity. So factors affecting enzyme activity. So we have three factors. We have temperature, and we have temperature, pH, okay, and substrate concentration. Okay. Of course, by giving these three different factors, the manner at which the enzyme will react, they will react differently, okay, depending on what condition they're in. Okay, so whenever you are explaining these factors, okay, affecting the enzyme activity, it is very helpful for us to use diagrams. Okay, Okay, diagrams, or more importantly, the graphs. Because the graphs in temperature is different from the graph that is being applied in pH and in substrate concentration. So let's just say, for example, we have the graph for temperature. So each and every enzyme, okay, they usually work effectively in an optimal temperature. Since our enzymes that are present in the body works in the temperature wherein it is within the range of 37 degrees Celsius, then therefore most of them has to be in that condition. Otherwise, it can lead to denaturation. So how do we show that that explanation? So for example, this is 37 degrees Celsius. We have to indicate that this is the peak, okay, peak of the enzyme activity. So as the temperature increases, the enzyme activity also increases until it reaches a peak. This peak area, this shows that this temperature is when the enzymes work most efficiently. Okay, so more substrates are being changed into products. But when the temperature is going to be increased further, then it can lead to a decrease in enzyme activity because of the denaturation of the enzyme. Okay, so same thing with the pH. You have to include as well the explanation. Well, it is also helpful if we are able to make an annotation on the graphs that we are doing. Like for this one, you have to indicate that in this area, the reason why there is a steady increase here or a rapid increase here, because as the temperature increases, the enzyme activity increases. There is a decrease here because when the temperature increases more further, then it can lead to denaturation, therefore decreasing the enzyme activity. So by making an annotation on what the graphs show, then we are also training the students on how to read the graphs and of course teach them under what conditions the enzymes should be. Okay? So that will be all for enzymes.